Meiosis. Sexual reproduction is a form of reproduction that creates genetic variety. This requires the participation of a male and female of the same species. In order for successful fertilization, the male and female gametes must share genetic information through contact, creating chromosome pairs. There are a certain amount of chromosomes in each cell, varying between species. In humans, there are 46 chromosomes, 2 chromosomes per type. However, in the gametes of sexually reproducing organisms, there is only 1 chromosome per type, so that when it interacts with a gamete from the opposite sex and combines genetic information, the resulting offspring will have the appropriate amount of chromosomes. The way these gametes are produced is through the process called meiosis. Gametes began as diploid cells, and after having undergone meiosis, become a haploid cell. Meiosis is comprised of two main stages, meiosis 1 and meiosis 2. Meiosis 1 begins with a stage called interphase. In interphase, chromosomes are duplicated to create 2 identical sister chromatids in the beginning of meiosis 1. It is in this stage that crossing over occurs. Crossing over creates genetic variation by swapping certain parts of the chromosome with other chromosomes. This happens by literally crossing the chromatids, creating an X shape called the chiasma. There are generally between 1 and 3 chiasma per homologous pair. During this stage, the nuclear envelope disappears and synapsis, or the fusion of chromosome pairs, occurs, creating bivalence. The next stage of meiosis is metaphase 1. This stage is identical to the metaphase stage of mitosis. At this point, the centrosomes have lined up at the poles, creating microtubule spindles that force the chromosome pairs to line up at the equator of the cell. In the next stage, anaphase 1, the homologous pairs are separated and sent to opposite ends of the cell. In the final stage of meiosis 1, telophase 1, the chromosomes uncoil. The nuclear membranes are reformed, and the cytoplasm separates through cytokinesis, creating 2 identical haploid cells.
Now begins meiosis 2. Meiosis 2 begins with a stage called prophase 2. During prophase 2, the chromosomes condense and become visible, and the nuclear envelope disappears. The next stage is called metaphase 2. This stage is identical to metaphase 1, where the centrosomes form microtubule spindles that line the chromosomes up at the equator of the cell. After metaphase 2 comes anaphase 2, where the centromeres separate and chromatids move to opposite ends of the cell for division. In the final stage of meiosis, telophase 2, the nuclear membranes reform and the cytoplasm splits once again during cytokinesis. The final product of meiosis is 4 genetically varied haploid gametes.
Important terms used in this video. Bivalent. A bivalent is 2 homologous chromosomes. Centromeres are the centralized region joining 2 sister chromatids. Chiasma. The X-shaped structure formed between non-sister chromatids during crossing over. Chromatids. 2 identical DNA molecules. Chromosomes, a thread-like gene-carrying structure found in the nucleus. Crossing over, the reciprocal exchange of genetic material between non-sister chromatids. Diploid, a nucleus containing 2 chromosomes of each type. Gametes, reproductive cells. Haploid, a nucleus containing 1 chromosome of each type. Homologous pair, chromosome pairs of the same length, centromere position, and stringing pattern that possesses alleles of the same genes at corresponding loci. One comes from the mother and the other comes from the father. Nuclear envelope. The membrane that encloses the nucleus. Synapsis. The pairing of replicated homologous chromosomes. Le fin. Belzenef. I feel like that was really dumb. Let's do something different.